Hi, my name is Daniel Morreale. First and foremost, I would like to thank Dr. Nigel Fraser and the selection committee for the Fraser Prize for selecting me for this honor. I'm excited to have the opportunity to share a little bit of my work today. My thesis work focused on an important pediatric pathogen, Kingella kingae, which is now a leading cause of bone and joint infections globally in children under 2 years of age. Additionally, K. kingae can frequently cause endocarditis or heart infections and sepsis or bloodstream infections. However, many more kids will be colonized by K. kingae than will go on to develop any of these diseases. We have very little understanding of the processes that allow K. kingae to leave the site of colonization in the throat and cause systemic disease. My work, entitled Defining Factors Important During Colonization and Invasion by the Pediatric Pathogen Kingella kingae, focused on better understanding this process. Work from the St. Gem Lab, as well as others, has identified a number of virulence factors that are important during the pathogenesis of K. kingae disease. My work focused on just one of these factors, which is secretion of an RTX family pore-forming toxin called RTXA. Toxin secretion is required for the ability of K. kingae to lyse host cells, and loss of the toxin results in an avirulent strain in animal models of infection. When we look at its broader bacterial family, only one close relative of K. kingae, Kingella negavensis, can also secrete RTXA. All other members of the bacterial family, the Neisseria, are non-toxigenic. This led me to ask the question, how was RTXA acquired by the pathogenic Kingella species? By looking at a large collection of isolates, able to identify a very unique pattern of evolution for toxin secretion by K. kingae. First, an ancestor of all of the isolates in the Neisseria evolved a type 1 secretion system to secrete a distantly related RTX protein called FRIPC. FRIPC is non-toxigenic and is not required for pathogenesis in any of these strains. Over evolutionary time, we had a number of speciation events eventually coming to the lineage that resulted in K. negavensis and K. kingae. Within this lineage, a secondary island was acquired, which harbored RTXC, RTXA, the toxin gene, and TOL-C. However, this island lacked the ability to secrete the toxin. In order to successfully secrete the toxin, this lineage had to co-opt the type 1 secretion system previously extant in the Neisseria. With that type 1 secretion system, this lineage is now pathogenic and able to cause invasive disease by secretion of RTXA. Finally, within the lineage that becomes K. kingae, we get a duplication of this island within the genome, resulting in 2 genotypes. Genotype 1 isolates have a single copy, genotype 2 isolates have 2 copies of RTXA. Genotype 2 isolates are associated with increased virulence, both epidemiologically and in vitro when we test them at the bench. Taken together, this is a very unique pattern of evolution for toxin secretion of an RTX toxin, and the acquisition of this island is essential to virulence within the pathogenic Kingella species, marking it as a key event in the evolutionary history of this lineage. Now that this lineage could secrete RTXA, it could more readily cross the epithelial barrier, causing the invasive disease that we associate with Kingella today. Thank you again to Dr. Nigel Fraser, the selection committee for the Fraser Prize, and my mentors, Dr. Joe St. Gem and Dr. Paul J. Planet at the Children's Hospital of Philadelphia. Thank you.